hybrid. Option 4 are there, none of these. Okay, now this is the question that asked in EMS also. This question same asked in JIPMER also. This question same asked in All India PMT 2014 also. So this is very important questions. I have seen this one, very important. Now come on, how we will solve this. In the question only given hybrid, I given this one so that you understand N15 and 14 means hybrid. This is not given in the question. What told? N15 and 14. Okay. This is N15, this is N14. Okay. Now they are telling that this is continuously kept in N14 medium thrice. Okay. So thrice means 60 minutes. So come on that. So this is N15, this is N14. Now in the first, what will happen? Here N14 and here also N14. Okay, this is the first. Now come on second. So second, what the things are coming? Here we see this is what? N15, this is N14, N14, N14. This is having. Now in the second, what happens? This also will N14, this also will N14, this also will N14, and this also will N14. Okay, if we are coming on third generation, so come on that third. So in the third, what will come? Here see, 1 in the one generation, how much will come? 2. In the second generation, how will come? 4. Okay, 1, 2, 3, 4. Now these 4 will give how much? 8. So come on that. So this is there, 1, 2, 3, 4. Here what happens? The heavy isotope is being removed. Why? Because N4Cl, that was the, it is density gradient, they are mingling that one. So here how much will come? Same. This one, it is N15 and 14. This is N14 and 14. Same. N14 and 14. Same. This also same. This also same. This also same. This also. So how much came? 1, 2, 3, 4, 5, 6, 7, 8. Okay. So total came 8. So how much heavy came? Heavy DNA. So heavy DNA was 0. Okay, heavy DNA how much? 0. Light, if we are seeing, so 1, 2, this also 3, 4, 5, 6, 7. So in the 8, 0. Light DNA came how much? 7, and 1 came what? This is hybrid. So hybrid DNA came how much? 1. Okay. Now, so what will be the answer? Where answer is there? What the options were there? Options were 7 light, 0 heavy, and 1 hybrid. So this is the correct one. So how much came? 7 light, 1 hybrid, and 0 heavy. Okay, this is hybrid, and remaining all came what? Light. Heavy already miscible. Okay, they are dissolved in the N14 medium. Okay, so this is the questions that mostly came in the medical exam. Now, Michelson and Stahl that proved that semi-conservative nature of replication. Now, if we are coming on mode of replication, okay, so what are the things that is coming on mode of replication? So come on that mode of replication. Mode of replication means how it started, what are the steps involved. Okay, so in the mode of replication, what happens? A region in the DNA where replication starts, that is called origin of replication, or ORI. Okay, so what I told, a region of DNA where replication starts or duplication, because they are making exact copy of their parent DNA, so that's why duplication. So a region of DNA where replication starts or duplication starts, known as ORI, or full form is origin of replication. Now, in prokaryotes, this ORI is known as ORI-C. So, in prokaryotes, like what? Like E. coli bacteria, like E. coli bacteria, ORI is known as ORI-C. Okay, they are known as what? ORI-C. Okay, now prokaryotes and viruses has single ORI site. Okay, what I told, in prokaryotes and viruses, single ORI site is present, whereas in eukaryotes many ORI site is present. This you have to remember. Okay, what I told, that in prokaryotes ORI is also known as ORI-C, and in prokaryotes and viruses only single ORI site is present, but in eukaryotes many ORI site is present. Okay, now if a single replicating unit is there, that is known as a replicon. So what I told, single replicating unit is known as a replicon. Now questions are asking, prokaryotes contain one replicon, many replicon, 10 replicon or none of these, like that questions will come. 5 replicon, so you have to remember that single replicon present in prokaryotes, and in eukaryotes number of thousand, several thousand replicants are present. So in eukaryotes several thousand replicons are present. So now you understood that replicons are what? That is single replicating unit known as replicon. Okay. Now one more scientist came. That scientist name is Cairns. Cairns. So he proposed theta model for circular DNA replication. Okay, what I told Cairns. Cairns proposed theta model for replication of DNA. Replication of DNA. And also proved that T3TDR, what is that? Tritiated thymidine. Okay, tritiated thymidine for semi-conservative nature. Semi-conservative nature of replication of DNA. Replication of DNA. Okay, and where? In E. coli bacteria. It is not proved, it is supported. Okay, and also supported because it is already proved previously. So, and also supported the tritiated thymidine T3TDR for what? Semi-conservative nature of replication of DNA in E. coli bacteria. Okay. Now, this experiment done with the help of autoradiography. So it is supported. How much? How it is supported? This is supported by autoradiography experiment. Okay, this you have to remember what the technique was there. So autoradiography. Okay, now next we have to come. What are the things essential for DNA replication? So that is prerequisites for DNA replication. So come on that. Prerequisites for DNA replication. Now, what are the things are there that are required? First, we have to take. What we have to take? We have to take DNA template where the new DNA will form. So, DNA template. Now, another thing, first we already seen that DNA template, then what we have to take? DNA unwinding proteins, because this DNA is what? Having double helix model. This type of things are there. So it has to first unwind, then replication will start, duplication will start. So second thing is DNA unbinding or unwinding proteins. Okay, DNA unwinding protein. Another thing is what? DNA gyrase and helicase. DNA gyrase and helicase enzyme. Where that will be used, we will see. So DNA gyrase and helicase enzymes. Then what? RNA polymerase enzyme for what? Starting the replication by the help of RNA primer. So what I told? RNA polymerase enzyme. For what purpose? For synthesis of RNA primer, because this is an important one for starting the replication. Another thing, DNA polymerase enzyme 321. Three types of enzymes are there. DNA polymerase 3, 2 and 1. This also important, DNA polymerase 3 to 1 or DNA polymerase 1 to 3 enzymes are there. Okay, then what we need? Then we need deoxyribonucleoside triphosphate. Deoxyribonucleoside triphosphate. Okay, deoxyribonucleoside triphosphate. Then what activator we need, and what activator here acts? So magnesium ion Mg2 plus ion acts as activator. Okay, now what more things is there to join the fragments? Okazaki fragment. What is Okazaki? We'll see that. So DNA ligase, one enzyme is there, DNA ligase. So DNA ligase enzyme to join the Okazaki fragments. Okay, so these are the raw material needed for what? Needed for DNA replication. Okay, so these are the raw material that is needing where? For the DNA replication. Okay, now just for giving one, I am just giving one diagram, and then we will discuss in detail. So how it is going on. So this is the parental DNA. This is unwinding. So it is looks like what? Y. So that making what? Y fork. Now we already know this is 5 prime, this is 3 prime. So here also it will come 3 prime, this will come 5 prime. Okay, now this is the parental DNA. Here what I told, this is the formation of DNA takes place continuously. In which direction? This is 5 dash, this is 3 dash. Okay, now here also what will happen? I told that it is semi-conservative. So this is parental DNA and this is daughter DNA. Here they are forming in the discontinuous pattern. How it will go? This is 5 prime, this is 3 prime. Okay, so formation, what I told, formation always start from 5 prime to 3 prime, but on template 3 prime to 5 prime. Now here what we are seeing, that this formation occur continuously. Continuously. continuous. And since continuously it is forming, so this strand is known as a leading strand. Leading strand. Now, here it is what forming? Discontinuous, discontinuous, and it is also known as a lagging strand. Now, this discontinuous strand, it is known as Okazaki fragments that forming. These are known as this is Okazaki fragments. Okay. Now, what I told that for initiation they need RNA primer. So here it is came here. This is what? This is known as RNA primer, that initiating duplication or replication. This is RNA primer. So it is what? RNA primer. Now, always here also it will come RNA primer because every time they need. So here small, small, small. So everywhere it is asking, it is starting their synthesis with the help of what? RNA primer. So these all are what? These are what? These all are RNA primer. Okay, these all are what? RNA primer. Now, what I told that when unwinding occurs, okay, what happens? Unwinding occurs. So you have seen that a rope. So if you open the rope, they want again to wind. Okay, so they needs, because we unwind, so they need what? A single stranded binding protein. Okay, so here what happens? One protein came that is known as SSB. It is SSB. So it is single stranded binding protein. This doing what? They don't want to, again they don't give the permission to wind again. Okay, this is their nitrogen bases attached with what? Hydrogen bond. Here they are forming like that. It is coming, they are forming new daughter DNA here. So like that they are doing what? They are forming. Okay, now what we seen, direction of movement of this one is known as replication fork. This Y-shaped occur, Y-shaped structure, it is known as replication fork. Okay, now what the movement going on? So replication fork movement is towards upside. So they are opening, this one is going on, so they are opening like that. Okay, and the movement further forming. Okay, so here enzyme are used to unwind that one. It is known as helicase. So it is helicase. All the things we will discuss. Okay, so this is helicase. Okay, DNA polymerase third, this one, RNA primer is there that going to form new. So here what it is being utilized? DNA polymerase third enzyme is being utilized. Okay. Here also same thing will come, DNA polymerase third, because they are forming. So here also will come DNA polymerase third, here DNA polymerase third, DNA polymerase third, here also. So when the formation of RNA primer helps to DNA replication, what the enzyme is used? DNA polymerase third. And this Okazaki fragments is a gating seal. Okay, so this getting sealed by the help of DNA polymerase 1. So here DNA polymerase 1 is being utilized. Okay, now with the help of DNA polymerase 2, when replication ends, RNA primer will remove. So this removal occurs with the help of DNA polymerase 2 and 1 combinedly. Okay. So this is the what I told, Y-shaped structure replication for single stranded binding protein, Okazaki fragments. Okay. So these all we seen. Now we have to go on inside with the help of theoretical portion. This is what diagrammatically you understand that how replication starts. Now you have to understand that continuous formed newly daughter DNA known as leading strand. Here discontinuously formed newly formed DNA known as lagging strand. Okay. Now, always what I told, template strand will be 3 dash to 5 dash. Here also see 3 dash to 5 dash or 3 prime to 5 prime. And new DNA formation from 5 prime to 3 prime. Here also you see 5 prime to, this is 5 prime to 3 prime, 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 5 prime to 3 prime. Okay, so like that it is going on. Now, this scheme shown by Kornberg, scientist name was Kornberg. So come on that, what is that? This figure is Kornberg scheme for DNA replication. Okay, so this was Kornberg scheme for DNA replication. Now we have to come on, replication takes place, how the steps utilized. So come on that. DNA replication takes place with the help of following steps. Okay, now what are the steps are there? So the first step is activation of deoxyribonucleotides. What is that? Activation of deoxyribonucleotides. First step is activation of deoxyribonucleotides. What happens here? In the presence of energy and enzyme, one enzyme is there, phosphorylase. Okay, in the presence of energy and enzyme phosphorylase. This enzyme you have to remember. What enzyme is there? Enzyme phosphorylase. Four type of nitrogenous bases are there. So DAMP. Now, what is the full form of DMP? Deoxyadenosine monophosphate. Deoxyadenosine monophosphate. Another one, because we are discussing about what? DNA. So one is adenine, one is what? Guanine. So deoxyguanosine monophosphate, DGMP. Deoxyguanosine monophosphate. Okay, so first we seen DMP, next is DGMP, next is DCMP. CMP, what is that? Deoxycytidine monophosphate. Okay, deoxycytidine monophosphate, and the last one is and DTMP, deoxythymidine monophosphate. Now, these are what? So, in the presence of energy and enzyme phosphorylase, these four DMP, DGMP, DCMP, and DTMP converted into active triphosphate form are converted into active triphosphate forms. So what I told, DAMP converted into DATP in the presence of what? Phosphorylase and energy. Same D, second is what? DGMP. So GMP converted into GTP, triphosphate. DCMP converted into DCTP. DTMP converted into TTP, means this monophosphate converted into triphosphate. Monophosphate converted into triphosphate in the presence of what? Phosphorylase, and that process is known as activation of deoxyribonucleotides. Okay, now you have to remember one thing more. What? This DATP, it is what? Used as energy currency of the cell. Used in energy currency of cell. Now, this GTP used as protein synthesis, mostly it is used in this energy used in protein synthesis. Mostly GTP used in what? Protein synthesis. CTP used in phospholipid synthesis. This energy used in phospholipid synthesis, and TTP used in complex carbohydrate and polysaccharide synthesis. So it is used in complex carbohydrate and polysaccharide synthesis. These four you have to remember. What I told: ATP, energy currency of cell, the energy, okay? GTP, energy used in protein synthesis. CTP, energy used in phospholipid synthesis. TTP, energy mostly used in complex carbohydrate and polysaccharide synthesis. This is a very important. What are the used energy used for what formation? So this you have to remember. So first step